Okay, so for 17, in this chapter, we start to look how we go from the genes to the proteins. So the genes are going to tell your cell what proteins to make, and it does it through transcription and translation in there. Beadle and Tatum were some people that studied this process early on, and so remember it was in 1953 we had Watson and Crick that came up with the structure of DNA. So in 1958 they had this hypothesis that genes act by regulating definite chemical events in there, and it wasn't quite right in there. So they decided to revise the hypothesis a little bit to say that you had one gene led to one polypeptide. It's a little bit closer, but it's still not quite accurate in there. And so a lot of times with a eukaryotic gene, each is going to code for a set of closely related polypeptides in there because of something called alternative splicing in there. You can have one gene and different variations of how you take the pieces of that gene lead to different proteins in there. A lot of genes are actually just going to code for RNA molecules that they never go beyond that. Their job is to become an RNA molecule, and they have different functions as RNA where they can act like enzymes in there. So the process has actually been modified a little bit in there and what we thought over the years in there. So your DNA is going to provide the instructions for making proteins through an RNA. So if you're a prokaryotic cell, you start with your DNA and you make an RNA copy of it doing transcription, and then you use the mRNA to make a polypeptide through translation, and it all occurs in one cavity in there because that's all you have. If you're a eukaryotic cell, you're going to do transcription to make your pre-mRNA in the nucleus, and then it gets processed to become your mRNA in the nucleus. Then it goes out into the cytoplasm where translation is going to make your polypeptides from it. So this is in two separate compartments in there, which is going to make a difference in some of the things they're able to do in there. So transcription is just the synthesis of an RNA using a DNA template in there, while translation is making the polypeptide using the mRNA. Your mRNA is your messenger RNA, and it's going to have your genes, protein building instructions in there. It's going to carry the genetic message from the DNA in there to the synthesizing machinery in the cell. So the ribosomes are going to be the organelles that are able to actually facilitate putting together your amino acids into a polypeptide. So in a eukaryote, you're going to have the transcription in the nucleus, translation in the cytoplasm. In prokaryotes, it's all in a single compartment, so you can actually begin to do translation before transcription is done. You can't do that in the prokaryote because they're separated in there. So your pre-mRNA is your initial RNA that's transcribed prior to being modified, so it's sometimes referred to as a primary transcript. So when we look at the genetic code, you have 4 nucleotides that make up your genetic code, yet you have 20 different amino acids. So one letter isn't going to work in there to code for that. Using 2 of them isn't either. That would give you 4 times 4 is 16 possible combinations. Still not enough. So instead we have to do a triplet code in there. So you have 4 times 4 times 4 is 64 possible combinations of letters that will come with that. So your genetic code is actually wrote as a triplet or a triple code in there, where you have series of 3 nucleotides that get translated in the mRNA. So if you look at this here, where you've got the codon, these are in series of 3, and the codon is going to be the 3 base pairs that code for a specific amino acid. So when you've brought this down to make your mRNA, It's units of 3 that you're going to look at and actually read, and those 3 code for the amino acid, which is what's down here on this line. So your template strand is the DNA strand that provides the pattern to make the RNA transcript. And you've got double-stranded DNA, so for any particular gene, it's always going to be the same side of the DNA, but there are genes on both sides of that piece of DNA in there. And it just varies on which side that particular gene is located on. So the RNA is going to pair similar to the DNA, but you're going to use uracil instead of thymine. So in DNA, where you pair A and T together, in RNA you pair A and U together. So your codons are going to be written in the 5' to 3' direction in there, and so the codons are also going to be read in the 5' to 3' direction. So remember, your DNA strands run anti-parallel. One goes one way, the other one goes the other way, so you've got 5' to 3', 5' to 3' in there. So you're also going to have your synthesis and translation is going to go in anti-parallel directions like that. So 61 of the 64 codons are going to code for amino acids. 3 of them don't code for anything, and they're considered stop codons. You're always going to start at the first AUG. The rule is almost universal. There's a couple of things they've found that have a few slight variations in there. For a long time, we didn't even know there was anything that did have a variation in there, but we don't know a whole lot about those few rare variations in the genetic code in there. So by having that genetic code be nearly universal like that, it's very easy for them to take a gene from one organism that makes a protein. When you stick that gene in another organism, it's going to make that same protein in there. So that is one of the reasons why it is so easy to insert genes into things like that. So there is some redundancy in the code, but no ambiguity in there, which means, like, if you look here, so for like phenylalanine here, you've got 2, leucine's got 2, it actually has these other 4. So there's multiple codons that will code for each amino acid in there. So your reading frame is going to depend on being able to correctly read the message, and so you need to make sure that you always start in the correct place. So if we go back up to this picture here, so when we read this starting here, we get UGG, UUU, but if we started here, you have a different reading frame, and you're going to get different amino acids out of the deal. So you may have seen some of those things. The English teachers like to collect them with punctuation, where they'll have one sentence in there that if you put the period in 2 different places, it means 2 different things. Basically the same thing on here. If you don't read it correctly, you're going to get different things out of it. So the good news is with this table is this is not something that you end up memorizing. So if you have to be able to read a genetic code in there, somebody would give you the codon table to do that with. It's just, you know, you don't memorize it. There's a few of them I have memorized from putting them on the board a million times that it's just easier for me to memorize it than it is to have to go look it up all the time in front of a class. But that's the only reason I have those memorized. So start is right here with AUG. And the way this works is you look at what the first base pair is, the letter along the left. And then your second base pair up across the top. So if you look and you go, okay, we have CC. That's going to bring you to this box here. And then you can go over here and go, okay, CCC. And it tells you what the amino acid is. Usually once you get to this box, you only have 4 choices. So some of them won't even include this column over here because you only have 4 to look through to find out what it matches. Like, I don't remember on that test right now. I've been trying to get all this stuff graded for the micro class. I told myself I'm not allowed to buy any Cadbury chocolate until I have all of their labs graded. Yeah, so right now I don't get any of those till I get all their stuff graded, and I'm really wanting my chocolate. So every waking minute that I can, I'm grading their stuff, and it's leaving me a little delirious in there. They need to go in somebody else's freezer because it can be frozen solid and I will just warm that sucker up and eat it. They're just not safe in my house. So when we look at the molecular components of transcription, you've got your double-stranded DNA. And when you are trying to make a duplicate of DNA to stick in another cell, you use a DNA polymerase. You're polymerizing it to make more of it, and you're making a DNA. That's why it's a DNA polymerase. Here you're going to make an RNA from it, and that's why it's going to be an RNA polymerase in there. And that's the easiest way I have for keeping track of. Is it a DNA or an RNA polymerase you use here? So what the RNA polymerase is going to do is it's going to split open the two strands of the DNA in there, and it's going to kind of hold it open in there. And the RNA nucleotides are going to be read in the 5' to 3' direction in there. So that 5' to 3', you just kind of want to let that be a little phrase in your head that haunts you, because that's the way everything's read. 5' to 3'. So your promoter is going to be the DNA sequence where the RNA polymerase is going to attach to it and initiate transcription. So it's going to help in the regulation of it. It also has to have a primer where it starts reading. So just like where with your duplication in there and needing to have primers, you have to have primers to get everything started. The terminator is going to be the signal sequence at the end that's going to tell it when to stop. And there's a little bit of a difference the way they do the terminators in prokaryotes and eukaryotes. So your transcription unit is that stretch of DNA that's going to be transcribed into the RNA. So bacteria just have a single RNA polymerase they use for making any kind of RNA in there. Eukaryotes have at least 3 different types of RNA polymerases. In your notes, I did put down the types that we know in here. However, as long as you know it's an RNA polymerase, I'm not going to expect you to know, is it 1, 2, or 3? My logic in that is we don't know that that's all of them. We might find slight differences in them. And then we have like a 2A and a 2B and everything else. You don't want to have to unlearn it. If you know it's an RNA polymerase, you've got the big picture in there. So when you make that RNA transcript, you're going to have your starting point in there where the synthesis actually begins, and the promoter is actually upstream from that. So here you've got your gene, you've got your starting point, and then you've got your promoter that's ahead of that you can see down at the bottom. In eukaryotes, you're going to have these other transcription factors in there that are going to help mediate the binding of the RNA polymerase and initiate the transcription. In prokaryotes, the RNA polymerase just binds onto the promoter. So then you have your transcription initiation complex, which is the transcription factors, the RNA and the RNA polymerase bound to the promoters. So in your promoter, one of the things they found is there's this DNA sequence in there that they call it a ta-ta box in there, that contains a bunch of TA sequences, about 25 nucleotides upstream from transcription for the start. So somebody must not have wanted their name on something that they called it a ta-ta box instead of whatever their name was. So you've got your transcription unit here, and your promoter, your RNA polymerase on there. So when it's initiated, you've got the strand unwound a little bit in here, and you're going to start this RNA transcript in there. And as this is going to move along your RNA polymerase, it's going to continue adding on to the 3' end of the transcript until it gets to the end. The RNA polymerase leaves to be recycled in there, and then you've got your completed RNA transcript there. So that would be your transcript. So the RNA polymerase will untwist the helix and expose 10 to 20 nucleotides at a time. Only adds to the 3' end, and then it starts to recoil afterwards. So in a eukaryote, it will occur at a rate of about 40 nucleotides per second in there. So a single gene can actually be transcribed simultaneously by several RNA polymerases. So as this one is done here, you can actually have another one that's coming along right behind it. That way you can send out multiple copies of those instructions. So in bacteria, the transcription has to proceed through a terminator sequence in the DNA, and that's going to function as the termination signal in there. In eukaryotes, the RNA polymerase is going to transcribe this polyadenylation sequence in there to make the pre-mRNA. And it does that about 10 to 35 nucleotides downstream before it cuts it off. So at the end, you have this polyadenylation sequence in there. So when you have this pre-mRNA, it has to be processed and cleaned up in there. And so we have to clean up the ends on this. So at the start end, you've got this 5' cap on here. That's going to be modified form of guanine on there. And it's 20 to 40 nucleotides in there. It's going to help to protect the end of the mRNA, and then also help to facilitate getting it where it needs to be in there. At the other end, you have this poly A tail in here. So once you get to the end, you have an enzyme that's going to add between 50 and 250 more adenines onto it. So by creating this tail, it's kind of got this piece dragging that helps to physically create some tension and pull it away from the DNA in there. So it's kind of like if you had a tail and somebody was like, hey, I don't want you to go over there, you could grab you by your tail and pull you back in the direction they want you. And then we're going to do this splicing in there, where large portions of the RNA that is initially synthesized is going to get removed. So initially they thought, before they sequenced the human genome, they were expecting to find about 100,000 genes in us. Then they sequenced the human genome, and they're like, oh, it looks like we're at about 30,000. And then in looking at it and starting to understand more what the DNA does, now they think we're down at about 10,000 genes in there. So we're not really as fancy as we thought we were in there. The genes are only about 1.5% of the DNA. So you do have lots of these other regions in here that regulate it. So you've got what we call these introns, which are intervening sequences, and then the exons are the expressed sequences in there. And so you can actually remove the introns, and then you splice together the exons, and that's what makes your coding segment. But what can happen with that is, say you've got a gene here, and we're just going to put 3 segments on this one. So when you're doing the splicing, you can say, okay, well, we're just going to use A for this one. Next one is going to be made of A and B. You have one that's made of A and C. A, B and C. B and C. A or B only, C only in there. So you can see where having just 3 parts can lead to 1, 2, 3, 4, 5, 6, 7 possible combinations in there. Most of the time you're going to have more than 3 parts. So the number of combinations that you can make, that's why we don't actually need as many genes as we thought we did in there. So, of course, all of these little spaces between in there and how they hook got named. So we have the SNRPs in there, which are the small nuclear ribonucleoproteins in there, which are the nucleotide sequences at each end of the intron. So they're used as a signal for RNA splicing, so it knows when to splice in there. The small nuclear RNA are in a SNRP, and each small nucleotide RNA is about 150 nucleotides long. So the spliceosome is going to be several SNRPs joined with additional proteins that interact with sites along the intron and help to determine what's going to be degraded and what's going to join for exons in there. So I'm not too worried about you knowing like what makes up a SNRP, a small nucleotide RNA in there per se. As long as, I mean, you will have seen the names and you know that there's these other regulatory sequences in there that are going to decide how you recombine these and how you cut them up. So some of the RNA molecules never get turned into proteins. They will function as enzymes themselves, and we call that a ribozyme in there. And so some of those introns aren't actually thrown away. They end up making ribozymes to catalyze other reactions in there, including its own exclusions. So one of the reasons that RNA can function as an enzyme is it's single-stranded, so it's going to allow it to have complementary base pairs in there to give it a 3D structure. And that's what it needs to do, catalytic functions in there. Some of them will contain functional groups in there that can participate in catalysis. And they're also able to hydrogen bond with other nucleic acids to add specificity to the chemical reactions in there. So the alternative splicing is this different combinations in there or different alternatives that you can get by splicing that pre-mRNA into the deal in there. So you can actually give rise to 2 or more different polypeptides depending on what you treat as an intron and what you treat as an exon. So the domains are the discrete structural and functional regions of the protein in there, the modular protein. So when you have exon shuffling in there, your introns increase the probability of having crossing over between the exons of the allele of a gene so that you can result in even more new combinations in proteins. So this just adds even more variation into the possibilities of an organism. So when we do translation in there, we have these tRNAs, these transfer RNAs, that are going to transfer your amino acid onto a growing polypeptide in a ribosome. So your tRNA has this region that is going to attach to an amino acid, and then it's got this other folded up region, and at the other end you have an anticodon. And the anticodon is 3 base pairs that are going to be complementary to the codon in there. And so they will be able to match up with the codon in there, be complementary to verify that you're actually bringing the correct amino acid down for what the codon wants in there. So this is showing it all folded up, but this picture tends to work better in there. You'd have an amino acid, you'd have the anticodon. So this would attach to something that is UUC in there. The table of codons is of the codons. So this is the anticodon that's complementary to those codons in the table. You would still have, because we don't actually read the anticodon, so you would have your UUC here, 5' to 3'. And that's going to come down in here, and it's going to have this amino acid attached to it, so that it will bring an amino acid that it will join with another amino acid in the chain, and then continue to grow. It's almost like, its function is almost like saying, okay, the tRNA is a box that you're going to put that amino acid in, and the anticodon is the label. So if you're following the instructions and it says go get box labeled ABC, you can look and go, okay, this is box ABC that goes with this step of the instructions. So it helps to make sure that you get the right amino acid incorporated in here. It makes it less likely for a mistake to occur. So the amino acyl tRNA synthase is a family of enzymes that are going to be involved with matching up the tRNAs and the amino acid in there. So we have 20 different synthases, one for each amino acid. And it's able to bind to all of the different tRNAs that are going to match up for that particular amino acid in there. So we have the 64 codons in there. So one of the things we know is we only have about 45 different tRNAs in there. So some of the tRNAs are actually going to be able to bind to more than one codon. And if you look at the codon table, a lot of times it's just the 3rd base pair that is different in there. So that allows it to have a little bit more flexibility in that 3rd site in there, so that you can use the same tRNA for multiple codons in there. So when the synthase catalyzes the binding of the amino acid on there, it's going to hydrolyze an ATP, and that's what's going to give it the energy to be able to do that. So we call it wobble, where you can have flexible base pairing in the 3rd position in there. So if you were to go back and look at a codon table in there, CCC and CCG both code for a proline in there. So if your first 2 are firm in saying, I must bind with a C, I must bind with a C, and the other one can be a little bit more flexible and say, I'll take a C or a G here. So your ribosomes have subunits to them. The rRNA is the ribosomal RNA that's part of the makeup of the ribosome. So the ribosome itself is made up of RNA partially. And what it does is it's going to facilitate that tRNA anticodon binding with the mRNA codon in there and make sure that things are matched up. So there is a little bit of difference between a bacterial and a eukaryotic ribosome in there. They function similarly and they have similar structure, but they're larger in a eukaryote than they are in a prokaryote in there. And so they measure them in Speedbergs, which is a sedimentation rate in there. So that's how you measure it, is how quickly it sinks. And that's actually really useful for us, because if you're going to try and kill bacteria in an organism that's a eukaryotic organism, such as a human, you would like to be able to kill the bacteria but not kill the eukaryotic cells. And by targeting specifically that bacterial type of ribosome, you're going to kill more prokaryotic cells. Occasionally it would be toxic to something like your mitochondrial DNA in there, which is going to be more similar because it's going to have the ribosome similar to go with from the endosymbiont theory where it was derived from the bacteria. But for the most part, that's one of the effective things about antibiotics and why they have worked so well is you can target it to kill a bacteria and not your cells. So each ribosome has 3 binding sites in it. So these things that look like little hot dog buns or hamburger buns here are your 2 subunits, and you've got the P site, A site, and E site, and they're actually in the order A site, P site to E site, so it's like a reverse ape in there. So the A site is officially called the amino acyl tRNA binding site. The easiest way to remember is it's the accepting site. That's going to be the one that's going to let the new tRNA come in. Then you've got the P site. This is where you're going to have the growing polypeptide. The peptidyl tRNA binding site's not, everybody calls it the P site anyway. You never see that full name of it used. So the way I remember it is growing polypeptide, and then the E site's the exit site in there. So what will happen is you bring one in, you attach the amino acid onto the chain. As it slides down, these 2 move down, and then one exits out the back. And it just keeps moving on down the line like that. So this picture is just showing them moving down the line. Here you bring one in to the A site. You attach the amino acid to the rest of them that are in the growing polypeptide site. And then it's going to move into the E site and exit out the back. So your translation initiation complex is the mRNA, the initiator tRNA, the small and the large ribosomal subunits. So all the pieces of it. So your initiation factors are the additional proteins that are required to bring that translation complex together in there. And then your elongation factors are going to be proteins that are involved in adding the amino acid onto the previous one. So it's always going to add it onto the C terminus in there. So it's always being synthesized from the N terminus to the C terminus, from the amine end to the carboxyl end in there of them. And then at the end you've got release factors in there. So the protein shapes in there, this is where it's going to basically come off, and then it will get folded up to go on its way with the release factors in there. So just like you could have multiple RNA polymerases reading the DNA to make multiple mRNAs at once, you can have polyribosomes in there where you have multiple ribosomes that are going to translate the mRNA at once. So here where you got the string of hamburger buns moving along the line, you can see that as it moves down it each gets longer in there. It's kind of like an assembly line. So if I do step 1 and then I'm done, I can move to step 2. Somebody else can start at step 1. You don't have to wait for me to be completely done. So after you make them, you're going to have your polypeptides start to foil or fold into their normal shape spontaneously. So if you remember when we talked about protein shape or the protein structure, you had the primary structure, which was the sequence. Secondary is where it made the alpha helices or beta pleated sheets. Tertiary, where it started to fold up. Quaternary, where you had multiple polypeptides come together. That's what's going to happen to them next. So your post-translational modification, after you make the protein, there might be some other steps that it needs to do to be modified to do its job. So free ribosomes usually are going to synthesize proteins that are going to stay in the cytosol. Bound ribosomes will tend to make proteins that are going to go into the endomembrane system and end up as secretions. So your signal peptides, these are going to be your marker polypeptides on proteins destined for the endomembrane system. They're going to help to get it there. You have the SRP, the signal recognition protein on there. These are escorts that are basically going to say, yep, I'm going to take you where you need to go. So some things that can go wrong here. Mutations, these are changes to the genetic information of a cell or a virus in there. This is going to help create more diversity in the genes. So a point mutation is going to occur at just a single nucleotide pair in there. An example would be sickle cell disease, where you had just a single nucleotide that mutated in there and you ended up with a disease. So smaller scale mutations, you can have a nucleotide pair substitution. You're going to replace one nucleotide in its partner with a different one. So sometimes that's not going to be a big deal. So say you had CCC and you replaced it with CCG, both code for proline. You replaced one nucleotide in there. So we call that a silent mutation because there's no observable phenotypic changes in there. It's going to make the same protein. That's usually your best outcome in the scenario. It's possible you could make an even better protein, but most of the time you're going to make something that doesn't function in there. A missense mutation would be where you change one of those amino acids in there and it puts in something different. So maybe instead of making it turn from CCC to CCG, you made it go from CCC to CGG or CGC in there. That's going to make a different amino acid in there and a different protein. The nonsense mutation, that's going to cause termination of translation. It's going to cause it to stop early. So that's where you would actually change it into a stop codon. Sometimes you'll just insert a nucleotide in or delete a nucleotide, and what that leads to is a frame shift mutation in there. So here, where you add in or remove one. So if you look at it here where you're normally reading along, you would go, okay, codes for MET, lysine, phenylalanine, serine in there. If we took out a letter in here, this U ends up missing. What you're going to read are different codons all the way down the line from there. You've moved your frame, you've shifted the frame that you read them in. And so those will cause big changes in there. Usually it's not going to end up being functional at all. So mutagens, these are physical and chemical agents that interact with the DNA and cause mutations. Sometimes you can have spontaneous mutations. These are just going to be created by, it's like a typo. You're going along reading it and you just misread it in there. It's estimated that 1 in 10 to the 10th nucleotides ends up being altered and the change gets passed on to the next generation. And there. Some examples of things that cause mutations: X-rays, UV light. There's a lot of different chemical mutagens in there. Xylene, formaldehyde, gasoline. It's just all kinds of stuff out there. So carcinogens are cancer-causing substances. Generally, they are a mutagen that's known to cause a mutation that leads to cancer in there. So let's go ahead and take a few minutes break here. There are a couple of videos that are embedded in the notes here that are actually going to do a really good job of showing things in here. So I'm going to skip over them in the notes because a drawing just, it's one of those things of videos like 1,000 words, and the drawing just isn't quite cutting it there. So what this looks at in there is how do we turn these genes on and off in there. You don't want to have genes going that you don't need to have running. You don't want to make proteins you don't need in there. So it's a way for the cell to save energy, but also make sure it's only doing the appropriate activities in there. So what they've done is they've studied these in operons in bacteria in there, and we know that there is a good likelihood when we study these things in bacteria, it's usually very similar that carries over into how it's going to function in a eukaryotic cell. So the operon has these different parts in there that are going to come together to be regulated. And the operons are either going to operate as default on or default off. And other things that will come into the environment are going to turn it on or off. And it's never just as simple as turn the switch on or turn the switch off inside of a cell. We know that when you're looking at the chemical reactions in there, you usually have a cascade of a couple things need to come together to turn it on, or a couple things need to come together to shut it off in here. So there's two of them that it will show you in there. There's the tryptophan operon in there that is going to be used as a repressible operon that's on by default but gets shut off. And then the inducible operon example it gives is the lac operon in there. So these DNA tube videos are going to be really good at showing you what happens in there to explain those. So another thing that can be done is regulating the chromatin structure. And one way of doing this is with attaching acetyl groups onto the lysines in the histone tails. We call it histone acetylation in there. And when you put these lysines on there, it's going to cause it to change shape so that the transcription proteins have easier access to those genes in there. So another way of doing that is attaching methyl groups on there. And so they talk about doing DNA methylation in there and also adding phosphate groups in there. So most plants and animals and even fungi are generally going to have more methylated regions be less active and the less methylated regions be more active. And these patterns can be passed on to successive cell generations to create some genomic imprinting in there. And so some of these things, like with the epigenetic inheritance, can actually explain why you can have two identical twins and only one of them get a certain genetic disease in there, is how the gene gets turned on and off in there. So the transcription factors, those proteins that need to bind on for transcription to occur, we have several different of those in there. You have general and specific transcription factors in there. General ones are essential for transcription of all protein coding genes. They have a low rate of initiation, where specific transcription factors are set for a particular time or place in there. And so they're going to have a more limited interaction for control in there. So when we silence a gene, that's a repressor that's going to involve additional proteins to deacetylate the histones in there to reduce transcription. So you can also bend DNA in there. So with that, if you bend the DNA in here, it brings these mediator proteins together to allow regulation for turning the genes on and off. So the alternative RNA splicing, this we already talked about in the other section in there, where you can have different sections of the exons come in so that you get different RNAs in there. It's estimated at least 75% of the human genes with multiple exons do undergo alternative splicing in there. So these proteasomes, these are protein complexes that are going to recognize these proteins that are tagged with ubiquitin and lead to degradation in there. So mutations can make specific cell cycle proteins become impervious to proteasomes, and they don't degrade like they're supposed to, which can lead to cancers in there. So I'm skipping through because there's some of it that I just feel like is more important than others, and we don't have huge amounts of time. So when you have a zygote in there, you've got two different people's genetic information that comes together, and you start out with a zygote that is one cell. And if you just cloned that one cell over and over again, you wouldn't be able to differentiate them into the different types of cells that make up a human in there. So differentiation is the process that allows these cells to become specialized in structure. Morphogenesis is the creation of form. So if you're studying morphology, you're looking at the form of something. And this is how the cells are going to become organized into tissues and organs in there. And so you've got these cytoplasmic determinants that are substances in the eggs that are going to determine the course of early development in there. So induction is when the growth factors and signals from the neighboring cells are going to cause a change in the target cell in there. Yeah, that when it's all of them in there, even if it's below or above it. So one of the things that we need to have happen in there is when you have this egg, you need to determine the polarity of it, a top and a bottom of an egg in there. And so they studied this in flies in there, and what we have are these bicoid genes in there that says the name means two-tailed in there, but it's used for determining the anterior and the posterior end in there. And so if the bicoid gene is not regulated properly, something could develop with two tails. Now, you're not going to see a human that's going to end up with two butts coming out of it and there for tails. That much of a variation is not going to be viable in there, but you can see it in some of the simpler life forms in there where you can end up with, instead of it developing a head and a tail end, it develops two tail ends before it ends up dying in there. So the things that are probably the most interesting to people are the oncogenes in here, which are the cancer-causing genes. Proto-oncogenes are the normal version of the gene that's going to code for the normal cell growth and division. That's what you have before it becomes an oncogene in there. So the big thing they would like to understand is what is turning these proto-oncogenes into oncogenes in there. And we don't have an answer to that. There's lots of things in the environment that can potentially do it. And sometimes it doesn't do it the first time through. It's with repeated exposure in there. It's not as simple as saying, okay, I'm never going to be around this, and it's not going to happen. You're exposed to all kinds of carcinogens in the environment every day. You may look at it and say, well, then I'm never leaving my house. Well, you stay in your house, and there are substances in your walls that come out of your house that are carcinogenic. And you say, well, then I'm going to go live outside in my yard. Well, UV light is carcinogenic. It's just you can't get away from it in there. So we have tumor suppressor genes in here. These create products that inhibit cell division in there. So they can help prevent uncontrolled cell growth in there. And these would be on by default. And so if something happens to one of those where they get turned off, then when it's not functioning, it can allow that uncontrolled cell growth to develop into cancer. So we have a few genes in here that we know will contribute to some cancers in there. The ROS gene in there, it stands for rat sarcoma in there. It's mutated in about 30% of human cancers in there. So those G protein relays where we talked about cell communication last time, in there, it will interfere with the G protein relays in cell communication in there. The P53 gene, this is a protein made from a tumor suppressor gene that encodes a specific transcription factor in there, and it's going to promote synthesis of cell cycle inhibition proteins in there. This is mutated in over 50% of human cancers in there. So that P53 protein functions as an activator for several other genes, including DNA repair. So the cancer model in there is, when we look at it, it's generally going to take more than one somatic mutation to produce all of the changes that are characteristic with a cancer. And that's why cancer incidence increases with age, is just as you get older, you accumulate more of these mutations. Yeah. And so some of them that have been studied more than others, the breast cancer gene in there is probably the one that's been studied the most in there. I mean, it's not all things are equal in women's health in there. There's some places where they're behind in women's health. But when it comes to a cancer being studied, breast cancer probably by far takes the cake in there and being studied the most. Everybody likes boobs, apparently. They're just, it has more research behind it than just about everything else. It's on the same gene in there. It's just, you know, there's all these foundations and all of this publicity for breast cancer in there. If, you know, there is all kinds of help groups for women, there is programs to help women with low income get cancer screenings and everything. If you're a guy with testicular cancer, nobody wants to help you. So in terms of that, women really can't complain on that front. We do have the opportunities for help on that type of thing a lot better than guys do in there. Heart disease, another story. It's been studied in men more than women, but you know, it is what it is in there. So the breast cancer genes in there are actually tumor suppressor genes that protect against cancer, and if they become damaged, then they no longer are protecting you against the breast cancer in there. So that's why if you have the gene in there, it doesn't necessarily mean that you are going to get breast cancer in there. So they know risk of cancer is lowered by minimizing exposure to DNA damaging agents in there. There are tumor viruses that can cause cancers in various animals in there. So they figure viruses play a role in about 15% of human cancers. Epstein-Barr virus in there that causes infectious mono is linked to multiple cancers, including Burkitt's lymphoma. So usually most people get the Epstein-Barr virus by the time they're a teenager in there. It's one of those things. If you were told as a teenager, don't date the opposite sex, don't kiss anyone, don't do anything like that because we don't want you potentially getting mono that's going to cause cancer later in life, are you going to listen? No. None of us listen when we're that age in there. So it just, it tends to get spread around. The HPV virus for cervical cancer, that virus will also cause cancer in any other tissue that it gets in. It likes the cervix, but it also likes the penis, the anus, and the throat. Right on. So, a lot of times males think, well, it only affects females. No, it will affect those things too. It's just, it's had more attention in females in there. Well, there are some things that when you have wrong with you, you don't really want to tell anybody. And, most men, men are not real big on talking about their problems like that. And if a woman has breast cancer, she usually tells everybody she knows. Everybody she works with is going to know. If a male has testicular cancer, there is a lot more social stigma in there of, oh, he's going to be impotent. He is going to be Mr. ED afterwards in there, and he's not going to want to talk to anybody about that. So it's just not as socially acceptable for men to talk about those things, and we will tend to really underestimate the number of males involved with it. Males are less likely to go seek health care for it. In there, you see support groups where women will get together and they will talk about things like that. How many male support groups do you know of? None. Yeah, there are pretty much none. But it doesn't mean it doesn't happen to them in there. So it's just the cancers for the females tend to get a lot more attention and a lot more money going to it. The biggest attention was Lance Armstrong for the testicular cancer, and everything with that kind of, went away in there with his, I can't even remember the name of it now. But, there's the Susan Komen thing, and you see thousands, hundreds of thousands of people that will go and participate in that. Nobody does that for the other stuff. The walks, they have 5K walks, everything. And when you go there, everybody is, assuming in there. They come up to me and I've been asked how my double mastectomy went at those. Because everybody just assumes, okay, yes, I am built like I have had that. I realize that. But, everybody just assumes in there, oh, let's coddle these women with cancer. And I did do one of the things, a bike ride, that was for, I can't even remember Lance Armstrong's thing, something alive or Live Strong. Yeah, nobody was walking around talking about testicular cancer or anything. It was, oh, Live Strong. There was no touching testicles, no looking at testicles, any of it. It's just a completely different approach in there. So it's just, well, I think it's socially unacceptable to ask me how my mastectomy went, but, it happens in there. So the research on the cancers in there is different, but also with it having multiple steps to get to a cancer, the idea that you're going to find one thing that cures cancer, it wasn't one thing that caused it, one thing isn't going to cure it in there. So, let's see.